Yeah. Yeah. Yeah, because it, apparently there was a whole outage for the whole system. So a lot of lectures, like not just for this class, for other classes, get cancelled. So they were able to recover partial file, and I posted it as IITS recovered. So you can listen to that. There's not much else I can do because the recording lectures is not mandatory. Okay, morning guys. Can you guys hear me at the back? Yes? Okay. All right, how was the reading week? Full of work? I agree. Anything interesting or exciting? Halloween? Anything else? Anyone watch the Jays game? That was not exciting. That was very sad. Sorry? Sorry, what did you say? It broke a lot of people's hearts. All right, so we'll get started. And a couple of things. Your grades will be posted sometime this week because I'm still waiting for 4 people to write the makeup of a makeup. So we'll have to wait and see. Okay? That's one. You guys also have a quiz this week, so don't forget, quizzes are starting back again. And lastly, there was an issue with Lecture 7 recording. Okay? I contacted IITS. They were able to partially recover the file, and you guys should be able to see that under the media, but there was a outage for Aura for a lot of classes. Okay? So I will not be able to have a recording for that because they just didn't record. It just didn't record properly. Okay? So in that case, just go over the slides, and if you have any questions, you can drop by during the office hours. Okay? Luckily, the lecture was a shorter lecture, so you didn't lose as much. But please go over the slides and come by during office hours if you have any questions. Okay? All right. Any other questions before we start? Everyone submitted their assignment? Yes? Okay. All right. So we'll start with our 9th lecture. And today we are going to be talking about retroviruses. And if we have time at the end, which we probably will, we'll talk about HIV. Okay? And just like we've seen with other viruses, we're going to look at the various genomes and their proteins, how these viruses replicate, what their life cycle is like. Okay? So retroviruses are viruses that have an RNA genome and they make a DNA copy of that genome. Okay? So it comes from reverse transcription. That's why it's retro, where you're making a DNA copy of an RNA molecule. Again, most of these viruses will have a spherical enveloped particle. Diameter is anywhere from 80 to 100 nanometers, and the envelope gets assembled at the plasma membrane. So what that means is some of the lipid composition, etc., will be similar to plasma membrane. It can have either an icosahedral or something we haven't really seen before is the conical capsid. And what we'll do is for most of these virus examples, we're going to be just focusing mostly on HIV. Okay? But one key thing about these viruses is that they have a reverse transcriptase. So in some cases, the reverse transcriptase is packaged into the virion so that it can actually carry out the reverse transcription reaction. So here's a false color image of HIV. And you can see it has a capsid that looks more or less conical. Okay? Here's a 3D model and the schematic with various proteins, etc. One thing you'll notice here is that it has 2 copies of the RNA genome. Okay, so you have 2 copies of the RNA genome, and you also have tRNA, a specific tRNA associated with this RNA molecule, or with these RNA molecules. In addition to that, you have an enzyme called integrase that's packaged in. You have your reverse transcriptase, and then you have your other proteins that we have seen before. So something we haven't seen before is 2 copies of the genomic RNA associated with tRNA, has an integrase, and has a reverse transcriptase. Okay? So retroviruses were initially classified into families, 2 families, and 11, actually they are reclassified into 2 subfamilies and 11 genera. Okay? So these are the various families here. This here got reclassified because it was discovered to be more diverse rather than just infecting simians, which are things like monkeys. Okay? So this information from the textbook is outdated if you have a 2nd edition. Anyone have a 3rd edition? I think a couple of people here do. So did they correct this in the 3rd edition? Okay. Because I don't have access to the 3rd edition. So can you please check and let me know? Thanks. All right. So this has been reclassified. So these are the various classifications. And if you really want to check, you can go to the ICTV website. And these are your various subfamilies and genera for these retroviruses. Okay? Now, these retroviruses are likely to be a relic of the RNA world. How do we know it's a relic of RNA world? Because they have a reverse transcriptase that generates DNA copies of the RNA genomes. Okay? Even though these viruses have a positive single-stranded RNA genome, it has a poly A tail, has a 5' cap. So why is it not just translated directly? Well, that's because reverse transcription needs to happen to make a DNA molecule. So these viruses have evolved such that even though the genome is positive single-stranded RNA, it still needs to be reverse transcribed. Okay? So it's a linear genome. It's a dimer of 2 identical molecules. Okay, so you can see that here, there's the 2 RNA molecules. The size is anywhere from 7 to 10 kilobases. That's packaged in the virions. And it's complex with your NC protein or the nucleocapsid protein. Okay? But once these viruses get inside the cell, they make double-stranded DNA from the single-stranded RNA genome. And the DNA needs to actually enter the nucleus. Okay? So the genome needs to get inside the nucleus along with this enzyme called integrase. What do you think integrase does? It directs the integration of DNA into the host cell DNA. Okay? So once that DNA is made, that host cell DNA gets modified where you insert this viral DNA. And as a result, this viral genome is now treated like a cellular gene. That's why it's difficult to treat conditions like HIV, because the genome becomes part of your genome. Okay? So if you look at the various proteins, etc., what we notice is that these retroviruses, they have a unique replication cycle. And that's based on reverse transcription as well as integration of their genomes into the host. Okay? Your reverse transcriptase produces DNA copy of the RNA genome, and your viral proteins get derived from these 3 genes, the GAG, POL, and envelope genes. Okay? And once these are synthesized, they are incorporated into the virions. So for example, There's a transmembrane protein and a surface protein. So TM and SU, that's the ENV or envelope genes. Then you have your matrix protein, your capsid protein, and nucleocapsid protein. That's called the GAG group. And lastly, you have your reverse transcriptase, you have a protease, and an integrase. So that comes from the polymerase group of genes, okay? But again, look at the structure of the genome. It has a cap, has a poly A tail, but it also has some other sequences which we are going to talk about in a minute. But again, you have these various genes, but all this needs to be converted to DNA before this can be used. So GAG stands for group-specific antigen proteins. So they are matrix protein, capsid proteins, also sometimes called the P24, and NC, which is your nucleocapsid protein. Then you have your envelope proteins, which is the SU is the surface protein, also called GP120. That just basically means glycoprotein 120 kilodaltons. And your TM is the transmembrane protein, which is also called GP24, okay? And lastly, you have your polymerase proteins, which are involved in genome replication as well as integration into the host. You have protease, which is PR, RT is reverse transcriptase, IN is integrase. And each of these are made as a polyprotein that gets cleaved into mature protein. So this is similar to what we have seen for other viruses, okay? So you still make the polyprotein, and then they get cleaved into mature proteins. So let's look at the structure of the genome and what these features are, and we'll go through one by one. But have you guys heard of reverse transcriptase before? Have you used reverse transcriptase before? If you used it, where did you use it? cDNA synthesis. Which course, or were you working in a lab? Working in a lab. So if you take, for example, BioD21, they do a lot of RT-PCR, which is reverse transcriptase PCR. So you isolate RNA, you make DNA, and check for gene expression, okay? So we can actually use purified reverse transcriptase in lab to make DNA copy of the RNA molecules, okay? All right, so let's look at these features. So 5' cap and poly A tail, you guys already know about that, so I'm not gonna go into detail. But on either end of the genome, both on the 5' and the 3' end, there is about 150 to 200 nucleotide repeated sequences, okay? And in this case, they're labeled R in this figure, okay? Next to that, there's a unique region that's called U5, or untranslated 5' region, again, anywhere from 800 to 200 nucleotides. And then you also have U3, which is the untranslated 3' region, and this is slightly bigger, 240 to about 1,200 nucleotides, okay? Now, within this, there's a specific cellular tRNA that gets attached to the genomic RNA by base pairing, okay? And that's where the PBS is, or the primer binding sequence, okay? And this is downstream of U5. So it's going towards the 3' end from the 5' end. Then you have Psi sequence, which is a packaging sequence for the genome. You have 5' and 3' splice sites. SS stands for splice site. And PPT is a polypurine tract. So lots and lots of purines. Which ones are the purines?
Purines would be like adenine and like guanine.
Okay, A and G, okay? But if this is RNA, what would be present instead of an A? Well, not necessarily. If you were to take that A and make a reverse copy, what would you get? So that's the question. Yeah. Are you going to get U? You're making DNA. So keep that in mind. Don't confuse these. So you have your polypurine track, which is A and G. And when that gets reverse transcribed, it's going to get transcribed to a complementary set of base pairs. So this is what's used during reverse transcription. Okay? All right, so what we'll do is we'll look at the basic entry mechanisms for these viruses. We have already looked at some of the proteins, but we'll go into a lot more detail of each of these. So generally speaking, retroviruses, they enter the cells through fusion. So these are enveloped particles, so they will fuse with the cell membrane. And the tropism is based mainly on the interaction of the surface protein with cellular receptors. So, for example, for HIV, you have CD4 receptor and a couple of chemokine receptors, CCR5 and CXCR4. Okay? So again, you have your binding to the receptors, followed by fusion. You get your capsid inside, reverse transcription happens, and then you transfer that into the nucleus where the genome is integrated. Okay? So the envelope either will fuse with the plasma membrane, or the whole particle will get endocytosed, followed by pH-dependent fusion. So again, this is similar to what we have seen before. You either fuse right at the membrane, or you can take in the whole particle, and then it fuses with the endosomal membrane. But either way, we are releasing the viral core into the cytoplasm. Okay? So how does that work? So again, there's a conformational shift in the surface protein that exposes an N-terminal domain that's hydrophobic. Okay? So the shift in this exposes the hydrophobic N-terminal of the TM protein, the transmembrane protein. And this is what gets inserted into the cell membrane. Okay? So during the early phase of infection, you include entry and reverse transcription, so making a DNA copy of the RNA genome, and then integrating it into the cellular genome. Okay? So the mechanism is a bit complicated because we are now adding an extra step as to first making a DNA copy and then integrating it into the host genome. And once the virus enters, the virus core gets released in the cytoplasm, and at that point, the reverse transcription can proceed. That's catalyzed by the enzyme reverse transcriptase, as we have seen before. And this enzyme is actually a dimer and has two major enzymatic activities. It's an RNA-dependent DNA polymerase. Okay, so it uses RNA as a template and makes DNA copy from that template. Additionally, it has RNase activities. It's RNase H. It's a ribonuclease. So what that means is it can actually digest RNA. And it specifically degrades the RNA part of an RNA-DNA hybrid that gets synthesized as we are making the copy of the genome. Okay, so we'll go into the detail of how this works. And your DNA polymerase activity is then facilitated by the viral-associated tRNA. So remember, when we looked at the genome, we saw that tRNA binds to both copies of the genome. And this tRNA actually acts as a primer for the DNA polymerase to start making a copy, start synthesis. Okay? And as you guys already know, retroviruses are associated with disease. So one major example is AIDS. And They can also cause other severe diseases, things like leukemia and various types of cancers in other animals. Okay? HIV and AIDS was first described in 1981, but the studies show that it originated back in the 60s. Okay? And from there on, it started to spread in the 70s. So there was a zoonotic spread, possibly from infected chimpanzee blood. So this has thought to be originated when people were hunting bushmeat, when they were hunting these chimpanzees and eating their meat or coming into contact with their blood. So just to show you guys an example, the cases worldwide have gone down quite a bit. This number was 3.2 million in 1995, and in 2022, it was down to about 1.3 million. Okay? And as you can see, it's prevalent worldwide. Obviously, some places have more prevalence than the others, and you can see that here. For example, Eastern and Southern Africa has 37% prevalence. Asia and the Pacific has 23%, and so on. Okay? So this is something that's present all over the world. And one thing we know about the HIV, or few things we know about HIV, is that it spreads through direct contact with certain body fluids. Okay? So the other person has to have HIV, and you have to come into contact with certain body fluids. So things like blood, or semen and preseminal fluid, rectal fluid, vaginal fluids, and breast milk. Okay? So this infected fluid must come in direct contact with bloodstream or mucous membranes for the virus to actually spread. Okay? So before, and there's still a lot of stigma about it. A lot of people don't understand that you can't really get HIV from things like kissing or hugging and sharing food, etc. Okay? But unprotected sex and so on, this can cause transfer of the virus. So for example, it can be passed from mother to the baby. Okay? If you are sharing injections, that can transfer HIV. Okay? And if you were somehow given contaminated blood. Okay? Now, if you look at the HIV virus itself, it's a type of a lentivirus, which means it works relatively slowly. So lentis in Latin means slow, so it works relatively slowly. The disease progresses relatively slowly. Okay? Now, the virus itself replicates and kills lymphocytes and macrophages. So it targets your immune system cells. Okay? So it replicates in these cells and then kills these cells as well. Now, there are different types of lentiviruses. You have your HIVs, you have your SIV, you have various types of other viruses as well for other animals. Okay? So we are going to be mostly focused on the HIV in humans. Okay, any questions so far? Yeah.
Why does it choose immune cells?
Sorry? Because immune cells have the right kind of receptors it can bind to. Always the case, right? Because it specifically binds to chemokine receptors which are present on immune cells. Okay? All right. So looking at the virus itself, it's a spherical enveloped particle, has a diameter of about 100 nanometers, and it has a conical capsid that has an icosahedral symmetry. So you can see the capsid here looks more like a cone than what we are used to seeing. Okay? It's a linear single-stranded RNA. It's a positive sense genome, and you have two identical genomic RNAs in each of the variants. Okay? In HIV specifically, you have a specific cellular tRNA. So this is tRNA for lysine, And these tRNA molecules are packaged in the virions, and they're used as primers for reverse transcription. So how do these tRNAs come into play? Well, first of all, when the virus is getting packaged, this tRNA will base pair with the RNA molecules, okay? And then it gets packaged. And once it's inside the cell, these tRNAs, they will act as primers for synthesis of DNA. And if you look at the infection rate, and once they look at the initial infection progressing to a full-blown disease, what we see is, first of all, you have your entry of virus into the host. Then it goes to its replication site or site of infection, okay? You replicate more and release virions into the circulation. Now, one thing that happens here is that your lymph node, the virus eventually gets to the lymph nodes throughout the body. And your body actually clears the virus from the bloodstream, okay? So there's an immune clearance of the virus from the peripheral circulation, but it continues to replicate in the lymph nodes, okay? Eventually, it will get to a point where it will start to destroy lymph nodes, release more virus into the circulation, and at that point, you get to the condition where you lose a lot of immune response. And as a result, you become susceptible to opportunistic infections, okay? So, like I said, HIV replicates in and kills lymphocytes and macrophages. And both these cells are CD4 positive. So what that means is they have a CD4 receptor on their surface. This is a glycoprotein that's found on the immune cell surface. So this is what the target for this virus is. And as the infection progresses, eventually you start depleting the CD4 positive T lymphocytes, okay? And as a result, the host becomes immunocompromised. And these patients then suffer from multiple opportunistic infections from other pathogens. And this is the main cause of death, okay? So for example, if your immune system can fend off or it can fight off something as simple as a common cold, in this case, that won't work. That common cold may progress to pneumonia or something else, okay? So this is a major problem because a lot of other pathogens that our immune system can normally fight off gets blocked here, okay? So, the immune system gets blocked, and as a result, those pathogens, they start to infect. Now, we can divide the HIV infection into multiple stages. You have acute infection, clinical latency, and finally, AIDS. So acute infection is initial infection, you get flu-like symptoms, and it also infects the gut-associated lymphoid tissues, also called GALT. And as a result, there's a significant drop in the CD4 positive cells. So let's look at the figure. So here's your viral RNA, and the blue line represents the CD4 positive T cells, okay? So initially, the viral replication will take place, and this is your acute infection where lots and lots of RNA is being made. What that means is you're making lots of virus particles. Your CD4 cells initially go down, but eventually the virus will become latent. So this is where you enter the clinical latency phase. Your cytotoxic T lymphocytes and your antibodies, they will respond to the infection just like they always do. But the virus replicates in lymph nodes. And as a result, it results in gradual depletion of the CD4 positive T cells. So you can see the blue line going down consistently, Okay, it's not a sharp drop, it's a gradual decrease, because sharp drop will look like this. Instead, what we get is a gradual decrease of these T lymphocytes. And again, after a few years, your immune system is compromised. You're replicating more and more viruses. So that results to acquired immunodeficiency syndrome, or AIDS. And at this point, there's high levels of virus in the blood, and patients can have lots of opportunistic infections. Okay? Now, within this, you can divide this further, because if this is left untreated, the disease can actually take several courses after an acute infection. So there are some individuals that are rapid progressors, about 10 to 15% of the population, that develop late-stage symptoms within 2 to 3 years. So this is where their immune system is destroyed fairly fast. Some patients are slow progressors, and that's majority of that. Okay, so anywhere from 70 to 80%, where the symptoms develop in 8 to 10 years. So it's not something that happens quick. It can take years for the infection to make you really sick. But there are some individuals that are non-progressors. So they are long-term non-progressors. Only about 5% of the people belong to this group, but they tend not to show any CD4 positive T cells decline in their body. Okay? So there's no decline in these individuals. Now, we've gotten to a point where antiviral drugs can actually control HIV infection. Okay? It can prevent disease progression, but there's no effective vaccine just yet. Okay? Because remember, even if you were to vaccinate, some of these virus particles will stay in the lymph nodes, and eventually it takes years for the infection to progress. By that time, your antibody titer is very, very low. Okay? So unless you're getting boosters every year, it won't really work very well. Okay? And sometimes you may not even feel the symptoms until it's too late. Okay, so that becomes another problem. There's another issue with this. Can you guys think of a reason why the vaccines wouldn't be very effective? Can you guys think of a reason? Yeah. Okay, so the virus DNA is integrated into the host DNA. So you are making more and more viruses over time. So even if you were to have vaccine, it won't be very effective because you keep generating new virus particles. Okay, so that's why there's no effective vaccine for HIV just yet. All right, so let's go back. We looked at the genome and the various features. Okay, so what we are going to do now is look at how all these features play a role in your reverse transcription. Okay, any questions so far? Yeah. Sorry, one second. This slide? Yeah. Mm-hmm. Yeah. Not sure what the exact mechanism is, but somehow their T cells don't decline. The CD4 positive T cells don't decline. So it's possible that they somehow have a suppressing mechanism, but I'm not sure about that. Okay? All right, so again, looking at these features, let's see how reverse transcription takes place. So again, remember there's a R sequence here, repeated sequences. there's a splice site, your primer binding site, and your U5 and U3 sequences, okay? And in addition to that, you have your polypurine tract, okay? All right, so how does this work? Well, first you need to make what's called the strong stop DNA. It's a minus strand strong stop DNA, okay? So remember, there's a tRNA bind to the PBS, the primer binding site. Now, this will be used as a primer, and synthesis will take place in this direction. So this, in this case, will be 5' and 3'. So this synthesis is 5' to 3', okay? So your first synthesis of negative strand DNA, it's going to extend from tRNA primer to the 5' end of the viral RNA template. In addition to that, you have your R plus U5, so it's going to basically continue synthesizing in that direction, as you can see here, okay? When it says strong stop DNA, what that means is, like, if you were to try to do this in vitro, DNA synthesis stops abruptly. So basically, somehow that sequence stops the DNA synthesis very rapidly if you were to do this in the lab. Now, again, you have a 100 to 400 nucleotide DNA copy of your R sequence plus your U5 sequence. As you can see here, it's labeled R' and U5', okay? Now, once this happens, once you made this negative DNA, you remove the template RNA from this site. So you see this green part here? See how that's removed in this step? So this is done by RNase H. So RNase H digests the RNA in this DNA-RNA hybrid. As a result, it frees your R plus U5 DNA sequences, okay? Now, let's review this very quickly. So you have your tRNA bound to the primer binding sequence. You synthesize your minus strand strong stop DNA, which includes this R and U5 regions. RNase H comes in, it deletes or digests the 5' end of the RNA genome, okay? But after that, we have what's called the first strand transfer. In this case, your DNA copy of your R and U5 transfers from 5' end to the 3' end of the genome, okay? And this is where the R sequences become very important, because your R sequence from this fragment will base pair or hybridize with the 3' R sequence here, okay? So here you can see the tRNA is now here. This is your R' and U5', base pairing here to the other R sequence on the 3' end. And at this point, you can start synthesizing full-length copy of the genomic RNA, okay? So this is done by reverse transcriptase, because you're making a DNA copy of the RNA, so you need RNA-dependent DNA polymerase, which is reverse transcriptase, and this will make the genome into a full-length minus strand DNA molecule, okay? And on either end is going to produce what are called long terminal repeats that have your U3 and R. U3, R, and U5. On one end, on the 3' end, you're going to make repeated sequences. So you end up with this long terminal repeat. Does that remind you of something else in our genomes? Let's see who remembers. What other sequences in our genome have these kind of repeats? Sorry? Telomeres. Okay, but that's for the entire chromosome. Okay. Anything else? Think something small. Telomeres are usually very large. Something small, something that moves around in our genome. Anyone? Yeah. Transposons. Okay. Remember, transposons have repeated sequences on either end. And we also have retrotransposons. Remember that? What do retrotransposons do? They involve a reverse transcriptase step. So where do you think that came from in our genome? From these kind of viruses, okay, that use reverse transcriptase. So during evolutionary time, some of those DNAs that got reverse transcribed got inserted into our genome, so they have become transposons. Okay? All right, so now you synthesize a full-length genome copy. So in this case, it will be the negative strand DNA, but then you also need to remove the template RNA from this. So again, RNase H comes in, and it digests all of the RNA except that polypurine tract we were talking about. So here's your full-length genome copy, and here you have your RNase H coming in, digesting the RNA, but it's leaving this polypurine tract intact. And this acts as a primer to synthesize positive strand of the DNA through reverse transcriptase. Okay? So first you use this as a primer, now you're using this as a primer. Okay? So you have your plus strand synthesized, again, as a strong stop synthesis. It gets initiated at the PPT primer, so your polypurine tract primer, and it extends through the newly formed long terminal repeats, along with the original tRNA, which would be about 18 nucleotides. Okay? All right, so you've done that now. Here's your synthesis. Again, you remove the tRNA and the primer using RNase H, which exposes the primer binding site on the positive strand DNA. Okay? So here is your RNase H again, and you see how this removes the tRNA, and it exposes that PBS sequence on the positive DNA. Now this goes and transfers on the other side and hybridizes with the PBS on the 5' side. So there's a second strand transfer, and this can expose PBS and hybridize with that. Okay? Now at this point, your reverse transcriptase now can extend both DNA strands. So what you get is a linear double-stranded molecule with long terminal repeats on either end. So this is called the proviral DNA. Okay? Everyone clear on that? Yeah? Okay, take me through the steps. Step 1. What's step 1? That's not step 1.
The tRNA binds to the PBS.
Nope. First, the virus has to get inside the cell. All right, that was a joke. All right, let's go through the reverse transcription process. Step 1. Strong stop DNA synthesis that you said earlier. What's the next step? Sorry? Remove some of that template RNA using RNase H. Then what? You have a strand transfer. Then what? That's the first strand transfer. Then what? You synthesize the full-length copy of the genome. Then what? Okay, you get rid of the template RNA again. using RNase H. Then what? Does it digest all of the RNA, template RNA, or does it leave something behind?
The polypurine tract stays there and acts as a primer.
Okay, your polypurine tract stays there, it does not get digested, and acts as a primer. Then what? You synthesize positive strand DNA. Then you have a second strand transfer. Okay? And then at that point, your reverse transcriptase can synthesize in both directions. You get a linear double-stranded DNA molecule with repeated sequences on both ends. And this is the proviral DNA. Okay? Everyone clear? Okay, let's take 10 minutes and then we'll continue.